In this video, we will discuss the histology of the liver. The word histology means the study of the tissues. A tissue, of course, is a group of interconnected cells with a similar structure and function. Each lobe consists of many lobules, and each lobule is a 5 to 6-sided hexagonal or pentagonal cylindrical tissue. So you can see here the liver, we've got the right lobe. If we focus in on this section of the right lobe, we see a series of lobules here, all connected together. So there's connective tissue between each of these lobules. And this one lobule here consists of these hepatocytes. Hepatocytes are hepatic cells, hepato meaning hepatic, cytes meaning cells. There are millions of hepatocytes in each lobe. Each lobe is about 0.8 to 2 millimeters in diameter, and there can be anything up to 100,000 lobules in the liver. This diagram helps us to understand the relationship between the liver, the lobules, and an individual lobule. So we've got one lobule. One lobule is interconnected with many lobules to then form a lobe. Different lobes form then the organ, the liver. This diagram shows the arrangement of liver cells into the cylindrical lobules. So if we take a look at this structure here, 1, 2, 3, 4, 5-sided, so this pentagonal cylindrical lobule, we can see that it's made up of this sinusoid. We'll talk a little bit more in detail in the next video about that. Individually made up of millions of liver cells or hepatocytes. It's got this branch of the hepatic vein in the center. So this is another lobule here. There is another branch of the hepatic vein. And then we've got this portal triad that consists of a branch of the hepatic portal vein, a branch of the hepatic artery, and a branch of the bile duct. And together these three vessels are called the portal triad. Between each lobule is a triad of the hepatic portal vein, a branch of the hepatic artery, and a branch of the bile duct, which drain into or out of the central vein, this intralobular vein. So intra meaning within, so this is the central vein or the intralobular vein or the branch of the hepatic vein, intra being within the lobule. The hepatic artery and the hepatic portal vein split into smaller vessels to form these branches, to form these arterioles or these venules, which run in between the lobes. And these together are known as interlobular vessels. So this portal triad is known as the interlobular vessels. Inter meaning between the lobes, intralobular vessel meaning within the lobe. Let's look at this in a little bit further detail using another diagram. So we've got the 5 or 6-sided shape here. We've got, you can see the hepatic artery coming from the aorta, supplying our oxygenated, oxygen-rich blood. We can see the branch of the bile duct. We can see the hepatic portal vein, and together, of course, that's the portal triad, the interlobular vessels. Then we can see connected between these interlobular vessels and the intralobular vessel, we can see these spaces here called the sinusoids. If we look at this diagram down here, here we have the arteriole, the branch of the hepatic artery, the venule, the branch of the hepatic portal vein, and the branch of the bile duct. Notice the differences in size. The lumen of the hepatic artery is much smaller than the lumen of the portal venule, or the branch of the hepatic portal vein. Both of those then form sinusoids. The sinusoid joins up, and therefore that allows the mixing of oxygen-rich blood from the arteriole and substrate-rich blood from the venule to mix in the sinusoid. Due to the high hydrostatic pressure at this end of the lobule, that forces the blood in this direction only. So blood flows in one direction from the interlobular vessels towards the intralobular vessel, the vessel within the hepatic vein, or the branch of the hepatic vein. And lining those sinusoids, we have hepatocytes. So individually, each one of those cells along that plate is a hepatocyte. The hepatocytes are organized into plates. They're separated by vascular channels called the sinusoids, and those form blood vessels linking the interlobular vessels and the intralobular vessels. The hepatocyte plates are 1 cell thick in mammals. Please pause the video and complete the diagram that you have. Let's take a look at the answers then. We've got the lobule, the 6-sided hexagonal cylindrical lobule. We've got the red structure here being the branch of the hepatic artery, the blue bigger structure, the branch of the hepatic portal vein, and the green structure here, the branch of the bile duct, and together the interlobular vessel. We've got then, if we focus in on that lobule, we can see along here the hepatocyte plate. We can see the sinusoid allowing the mixing of oxygen-rich blood and substrate-rich blood. We've got hepatocytes. You can see the names of the 3 vessels. And we've got the intralobular vessel, the hepatic vein. As we've said, the hepatocytes are the main liver cells, which are highly metabolically active, which can divide and replicate. The sinusoids are the spaces between the hepatocytes where blood from the hepatic artery and the portal vein mixes to increase the oxygen content of the substrate-rich blood, allowing hepatocytes to remain highly active. Here is an image taken using the microscope. You can see, if I use my pen, you can see along here the lobule. You need to be able to recognize from these images what you can see. We've got in the center of that lobule the intralobular vessel, the branch of the hepatic vein, and at each of the corners, just on the outside of the lobule, if we were using a microscope, we would be able to change the plane of this image, and therefore we would be able to make out the portal triad, the branch of the hepatic artery that supplies the oxygen-rich blood, the branch of the hepatic portal vein bringing the nutrient-rich blood, and the bile duct receiving bile from the bile canaliculi. Again, here's another image that you need to be able to label should you be given this in an exam situation.